Hello, everyone. Welcome to our Innovation Showcase. My name is Maria Bian, and I will be your host for today. Today, I will introduce our company, MaxWell Biosystems, and also our high-content electrophysiology platforms that you can use to functionally characterize your 2D and 3D neuronal cultures. All right, so let me begin. About MaxWell Biosystems, we are based here in Switzerland, and it is our mission to provide electrophysiology systems and platforms to all of our users around the world so that they can focus on their research in discovering new things in neuroscience and also to accelerate the development of novel drugs for brain diseases. Our core expertise include the development of microelectrode array platforms, both on the hardware and software point of view, and also we are experts in the applications of these microelectrode arrays. Our customers, as I mentioned, is all over the world, coming from academic labs, contract research organizations, biotech, and pharmaceutical companies. So here in this conference, we are hearing a lot about human iPSC-derived neurons in 2D and 3D cultures. And this is really cool that we have this technology right now because it is then possible to have human cells in vitro that are highly reproducible, patient-specific, and can be used for high-throughput experiments. In the past, we would need to take the tissue directly from humans, but now we can convert skin cells or blood cells into many different cell types, specifically neurons. And here we have two examples, for example, 2D cultures or co-cultures with astrocytes, and also 3D cultures such as organoids and neuron spheres. Now, with these two platforms, drugs can be tested in high throughput on human cells earlier, back in the preclinical stage, using this technology. And aside from this, of course, now there is a need for high quality readouts to characterize these cells and the effect of compounds. So this is where we come in. So typically, to characterize function of cells, the gold standard for electrophysiology is using patch clamp, where individually you have to approach the cell with a micropipette and detect activity from inside the cell. So this allows you to get really high quality signals from every cell. On the other hand, if you would like to see the morphology of the cells, how they grow and connect with each other, and also the different cell types, you can use imaging. Now, our technology is something in between. So we have high-density microelectrode arrays that allow us to get activity from each of the cells, similar to what you see here in the inset. So these are three activities of three different neurons. Plus, we can also use this technology for doing some sort of electrical imaging. So without using any dyes or any labels, we can detect the action potential initiating and propagating along the axons and get this activity. And from this, we can actually extract the morphology of each of the cells. So imagine you can do this for thousands of cells in your sample. So you can repeat this experiment also across multiple days. Now, how is this made possible? In these high-density microelectrode arrays, we have many of these tiny electrodes that are tightly spaced with each other so that these electrodes are able to detect activity not only from one part of the neuron, but from multiple compartments of the neuron, along the dendrites, along the soma, and along the axons. So that's why we can follow these very small signals propagating along the axons. So there are other microelectrode arrays that are low resolution. However, the advantage of having more electrodes in high resolution is that we can see all the cells, and we can identify the activity from each of these cells, whether there are 3 cells, but if you just have 1 electrode, you're not able to identify how many cells you're recording from. Also, every cell can be detected. This one can be missed in low resolution, while for us, we are always at the right spot because there are always electrodes everywhere. Plus, these electrodes are small. That's why we have access to the cell's small signals. With this, we can then read out at different scales, from the network level, meaning the whole population level, down to each cell level, the activity of individual cells, and even deeper to sub-cellular level, wherein we can really see the propagation of signals along these axons. So our platforms here at MaxWell Biosystems are these two. MaxOne, which is our single-well solution, and MaxTwo, which is our multi-well platform. For MaxOne, this is a really small device that you can place inside of the incubator during your recording. And each of the well has 26,400 electrodes, which I will explain in the next slide. For MaxTwo, the mainframe itself is a built-in incubator, so it has the top and bottom plate heated, and this cover closes, and you can have your cell cultures in the right environment during the recording. We have 6 well plates and 24 well plate format for MaxTwo. Now zooming in into each of the sensor for each of the wells, so this is the same for both MaxOne and MaxTwo. These high-density microelectrode arrays are here inside, where you can directly culture your cells, your 2D cultures or your 3D organoids. You can directly place them and use some coating and culture them here. So you can maintain it in culture similar to what you would do in normal plates. And here at the bottom, we have a 2 by 4 millimeter square area with in total 26,400 electrodes. So each of the well has this. That's why we have high resolution. We can get activity from every cell. So here, the distance between 2 electrodes is 17.5 micrometers. So here in this image, you can see that one neuron can be recorded by multiple electrodes at the same time. We have low noise readouts that allow us to follow these small signals propagating along axons. And also, any of these 26,000 electrodes can be used for electrical stimulation. So these are the neuronal assays that you can use for real-time analysis of the activity for each of your samples. I will go through them in the next slide. So these are some examples of what you can do. First, the activity scan assay allows you to be able to get kind of a full image of your sample on the device on each of every well. So on top, we have the fluorescence microscopy image of the same sample. So here you can see that there is kind of a hole in this sample. And at the bottom, this is the functional image that you can extract using either MaxOne or MaxTwo. So you can know the location of every cell and also how active they are. Here in this case, we're showing how the firing rate, so how many spikes per second there are for each of these cells. And you can see the same hole that is there on the fluorescence microscopy image. And you can imagine this is not just a single endpoint experiment. You can do this across multiple days. So you can actually follow how the network starts to form and also how the cells are starting to have the action potential and connecting with each other. Next, we have the network assay. So typically, to be able to detect if the network is starting to form, we observe these network bursts. So what this means is that each of these lines is actually activity of each of the electrode in the microelectrode array. So these are 1,000 channels in total, and each of the dot is one action potential. And whenever there is this burst, these lines or vertical lines that you can see, that means that all of the cells are active at the same time. And these are represented as peaks here in the network activity. So some cells have these oscillating activity, which is indicative that now the network is matured and all cells are interconnected with each other. And with this, you can use this to characterize different cell types. So you can also appreciate this in Sylvia's presentation later. And also you can use this to identify effects of compounds and in high resolution as well. So for example, if only specific cells are affected by a compound, these can be seen with this technology because some of the channels will be affected, some not. And then finally, which is actually our most exciting and also most unique feature in our assays is this axon tracking assay. So here you can extract the morphology of each of the cell, you can follow the morphology, this action potential propagation, and even calculate the conduction velocity for each of these axons. So then you can have a general overview of the velocity of the action potential propagation for your sample and compare it across different cell lines and also add compound and see how that changes. So with all of these possibilities, you can apply this in many different neuroscience applications. In 2D cultures, as what I have mentioned, not only for human iPSC-derived neurons, but also for primary neurons, for 3D samples such as brain slices, brain organoids, and neuronal spheroids, and as well as for retina. So we are going now to our next talk for today by Dr. Maria Sundberg from Sahin's lab at the Boston Children's Hospital in the US. So Maria Sundberg is an experienced scientist with a background in stem cell science and neurobiology. Her current focus is on modeling neurodevelopmental and psychiatric disorders with patient-derived iPS cells. Maria has received her PhD from the University of Tampere in Finland, and since 2014, she has been working with the Boston Children's Hospital Department of Neurology and Harvard Medical School. So during this time, she has developed and published various cell culturing methods to study patient iPSC-derived neurons in different neurodevelopmental disorders. So please help me welcome Dr. Maria Sundberg. Hello, Maria.
Hi.
Good morning. Can you hear us well?
Good morning. Yes, I can hear you.
All right, so we will put up your presentation and then you can start.
So I would like to start by thanking the organizers for this opportunity to give this talk today. I will be talking about functional phenotyping of neurodevelopmental and psychiatric disorders with human iPS cell-derived neurons. And first, I will start with the introduction of our disease model. So we study rare copy number variations of 16p11.2 in the brain development. Then I will talk to you a little bit about characterization of dopaminergic neurons and cortical neurons with 16p11.2 copy number variations. And finally, I will show you some new data of the functional axon tracking of the cortical neurons with 16p11.2 deletion. Microdeletions and microduplications of genomic region of 16p11.2 have been implicated in various childhood onset developmental and neuropsychiatric disorders like schizophrenia, bipolar disorder, and autism. Autism is more common in patients with 16p deletion, whereas schizophrenia and bipolar disorder are associated with 16p duplications. This genomic region contains 29 coding genes, and most of them are expressed in the brain. Also, brain imaging studies have shown that patients with the deletion have macrocephalic brain growth, which means increased cortical layer volume. Patients with duplication have microcephalic brain growth, which is decrease in the cortical volumes. These patients have severe symptoms. In autism, there are behavioral deficits, communication difficulties, social interaction deficits, learning difficulties, and lower IQ. Also, language development and repetitive behaviors have been detected in autism. In patients with schizophrenia, patients usually have psychotic behaviors, mood changes, social interaction deficits, and hallucinations. These are severe disorders without curable treatments, and currently there are no efficient drugs that can help with these. That is why we were really interested to study these copy number variations and genetic pathways that are related to these disorders to find new molecular targets for drug development. There is a little bit of an issue with this slide, but I'm going to hop on to the next one. So how do we want to study this is that we want to generate iPS cells that were CRISPR-Cas induced to contain 16p deletion or 16p duplication. And when we had these iPS cells that were isogenic, we differentiated these iPS cells into dopaminergic neurons, glutamatergic neurons, and GABAergic neurons in vitro. Then our aim was to characterize the disease phenotypes of these cells by looking at their differentiation capacity, electrophysiological properties, and transcriptional profiling. We wanted to identify molecular targets and test different drugs in vitro to develop personalized medicine for these patients. And our previous study was actually focused on dopamine signaling pathway that have been associated to be deficit in schizophrenia patients and autism patients. So we first differentiated the iPS cells into dopaminergic neurons in vitro. Then we plated the cells on the high-density MEA and looked at their development of their activity in the presence of astrocyte co-cultures. And as you can see on the panel B in the red, the 16p deletion networks have increased active sensors and frequency of active sensors compared to control populations after long-term culturing in the MEA. Also, the synchronization of these networks was significantly increased in the 16p deletion populations compared to control populations and compared to 16p duplication populations. Then we wanted to look in more detail, one of the pathways associated to 16p deletion, which is the ROA pathway. So in the patients with 16p deletion, when the KCD13 is reduced, the expression of RHO-A protein is increased as shown in the panel A. And then we wanted to treat these cells with a RHO-A pathway blocker, which is called erosine. And with this small molecule, when it was added for the cells after 5 days on the MEA, we were able to rescue the network activity. And we reduced significantly the number of spikes in the 16p deletion neurons with rosin, showing here in the panel B in pink compared to the red population. Also, we were able to reduce the weighted mean firing rate of the cells after rosin treatment, showing here in pink compared to the red panel. And then, of course, we wanted to look in more detail at the cortical development of these cells, because failures of corticogenesis are suggested to contribute various neurodevelopmental and psychiatric disorders. And deficits in cortical development have also been associated to these 16p deletions, as shown previously. And how we want to do this is to generate both glutamatergic and GABAergic neurons in co-cultures, because these are the main components of the cortical layer. And as you can see from this panel, during differentiation of the cortical development, there are several deficits associated to autism in the proliferation, neurogenesis, cellular migration and localization, and differentiation of these cortical layers. So in our hands, we used previously published protocols where we took iPS cells and transduced them with viral vectors that express NGN2 under doxycycline promoter and puromycin selection cassette. With this way, we can generate glutamatergic neurons that are really pure cultures. And then we generated GABAergic neurons by using a combination of ASCL1 DLX2 cassette under doxycycline promoter in the presence of puromycin selection cassette. And these GABAergic neurons were also really pure cultures that we can generate. And we can culture these cells for long-term in vitro. And before doing the transfections, we also confirmed that our iPS cells are pluripotent, as shown with this immunocytochemical staining in here, and they express pluripotency markers OCT4 and TRA160, and they have retained their copy number variation, showing in the qPCR panel P. In the deletion population on red, the genes of the 16p region were significantly downregulated compared to control or duplication population. Then we did the immunocytochemical characterization of the cells and identified that the IGABA neurons are truly expressing GABA, showing here in panel C, and somatostatin, showing here in the panel C under the. And the INGN2 neurons we characterized with the immunocytochemical staining of DPR1, which is a glutamatergic neural marker, and synaptic markers, synapsin and PSC95. Then we cultured these cells together and quantified number of DPR1 positive glutamatergic neurons and somatostatin positive GABAergic neurons. And also we verified that these co-cultures expressed synapsin between MAP2 positive and somatostatin positive cell cultures. And then we plated these cells on the MEA, and this is the low-density MEA from MaxWell Biosystems with 16 electrodes per well on the multi-well plate platform. We had a combination of control population with NGN2 and astrocytes, and NGN2 astrocytes and GABA cells, both in the Cas9 control population and 16p deletion population. And as you can see from the panel A and B, the weighted mean firing rate at day 40 was significantly higher in the 16p deletion networks with NGN2 and astrocyte co-cultures compared to the Cas9 control populations. And of course, in the presence of GABAergic inhibitory neurons, the activity of these networks was significantly lower. And same kind of phenotype we were able to detect on the number of bursts of these cells that was significantly increased in the 16p deletion NGN2 and astrocyte co-cultures compared to control populations or the co-cultures with GABA cells. And then we cultured these cells up to 100 days in vitro and measured again their activity in the presence of GABAsine. GABAsine is a GABA signaling blocker, so in the co-cultures of GABA when we block the kappa signaling, we can see significant increase in the 16p deletion INCN2 networks. And this is same levels in the 16p deletion INCN2 astrocyte culture, showing here in red and in pink. And similar kind of phenotype was also detected when we did a number of bursts analysis of these cells. So in summary, without GABAergic neurons, the INCN2 16p deletion networks are hyperactive. And when kappa signaling was blocked with cappazine, the new hyperactivity of the 16p deletion INCN2 neurons was detected. And this implicates that glutamatergic neurons are driving the increased activity and the bursting phenotypes of these networks. So that's why in our next experiment, we wanted to repeat the rosin treatment for the iPSC-derived NGN2 cortical neurons. Same way that I showed you previously with dopaminergic neurons, we were able to reduce the number of spiking and bursting with the rosin treatment in this culture. And interestingly, same kind of phenotype we detected with the 16p duplication cells, showing in the panel C on blue. And the rosin also reduced the activity of the 16p duplication cultures. Moving forward, we wanted to identify how these cells behave in the high-density MEA, and we used all the previously mentioned methods, activity scan, network characterization, network bursting analysis, network connectivity, and single cell analysis with action tracking tool. And why we want to look in this action tracking tool is that with these assays, we can identify new biomarkers and detect maturity and developmental stages of the cells in more detail compared to previous studies. And we can also study cell phenotypes, cell disease phenotypes, and rare copy number variations, and do disease modeling and drug testing for different copy number variations in neurodevelopmental and neuropsychiatric disorders. In our hand, we first did the action tracking for day 38 after plating the cells on the MEA, and did the spike sorting and identified neurons from this network. Then we repeated this assay for the 5 days time point of differentiation. And as you can appreciate from the picture, the development of the axons and axonal length and branching can already be seen differentiating based on the developmental time of the network. And here is a video of that cell on the day 38, showing the axon tracking tool. And here we are doing the axon tracking in the same neuron. I just added the videos here because I think they're really, really cool. And then at day 45 of differentiation, when we look the same plates again, you can see that the neurites and axons are really longer compared to the day 38 time point. So already after like 1 week, we can see a significant increase on the development of these axons. And then we wanted to look in more detail all various features of these cells and compare the axonal properties to 16p deletion networks. So to do this, we also recorded the axon tracking for the 16p deletion INCN2 networks. And I also want to mention that these cells were co-cultured with astrocytes. And compared to the control populations, you can already see from these pictures that these are more longer branches and more complex compared to control populations. And between day 38 and 45, there is a developmental difference and developmental maturation of the neurons and their neuritic branches. And here are some videos of the 16p deletion neurons. This is at day 38 of differentiation. And this is the same neuron with the axon tracking tool. And then at day 45 of differentiation, this was the more complex neuron with more complex neuritic branching and neuritic tree. And of course, then we looked this data in more detail and looked the neuron conduction velocity and analyzed it in all the wells and compared the different time points between day 38 and day 45 of differentiation. And based on this analysis, we could already see that the neuron conduction velocity increased during the developmental time. And there may also be differences between the control population and the 16p deletion neurons on their neuron conduction velocity. And to summarize our findings, this is the picture of the Cas9 control population at day 38 next to the 16p 11.2 deletion neurons at day 38. And we can already see here that the longest distance from initial site and total axon length are significantly different between these two different populations. And there is a clear disease phenotype that can be detected in the 16p deletion networks, which is really interesting. And currently we are analyzing this data in more detail and repeating these experiments on other MEAs. And also we verified that when we look the individual neuron firing rate from this axon tracking tool and total number of spikes, the 16p deletion neurons were significantly higher neuron firing rate compared to control population and significantly increased number of spikes compared to control population. And this data is consistent with the HD-MEA recordings that I showed in the beginning of my presentation. So we have verified that these disease phenotypes are repeatable across different platforms. So I would like to conclude my talk today. So I have been telling you that the 16p deletion glutamatergic neurons have increased firing rate and number of bursts compared to control neurons. And this data is consistent between the low-density MEA and high-density MEA recordings. And our axon tracking analysis shows that there are clear differences in the axonal activity of the 16p deletion cortical neurons, and this analysis of this data is currently ongoing. And next we will study synaptic marker expression and treat these networks with synaptic blockers to investigate in detail the synaptic connections of the networks. And these functional deficits probably play a real role in the dysfunctional cortical development in the patients with 16p deletions and causing neurodevelopmental deficits for these patients. And targeting these phenotypes in vitro will in the future facilitate establishment of functional drug screening studies. I would like to thank you for your attention, and I would like to thank our PI, Mustafa Sahin, all our lab members, all our collaborators, MaxWell Biosystems, Human Neuron Core, Flow Cytometry Core, and Rosamund Stone Standard Translational Neuroscience Center. And if there are any questions, I'm happy to take those.
Thank you, Maria. These are all super exciting new results that you're showing. Congratulations. And we have some questions for you. First from online, this is from Matt Kelly from Pfizer. For Maria, what was your ratio of GABA neurons to INGN neurons? Did you try different ratios?
Yes, that's a great question. Actually, I did try different ratios. I tried 60% of glutamatergic neurons with 40% of GABAergic neurons. But the data that I'm showing today was 80% of glutamatergic neurons with 20% of GABAergic neurons, because this is the ratio that we find also in the brain.
Thank you. And maybe as a follow-up on Matt's question, what differences have you found between the 60-40 versus the 80-20 ratio of the excitatory inhibitory neurons?
So when we have more GABAergic neurons in the network is overall more silenced. So we have to increase, when we treat the networks with GABAsine, which is our GABA signaling blocker, we have to increase the concentration of GABAsine in order to block the number of GABA neurons in the network. So that was the obvious difference I noticed. And of course, we want to keep using the optimized current setting that we have with the 80 and 20%.
Thank you. Do we have any questions from the audience? Any questions from our online audience? Okay, I have a question on the axon results. So we have seen the control versus the other, sorry, I forgot the name of the cell type, that the other one has longer axons compared to the control. Do you have any insights on why this is the case?
Yes, I think this is really great question. So I think the pathway that we are looking at, one of them, the KCD13-ROA signaling pathway, when it's disturbed in the 16p deletion neurons, these cells tend to grow longer axons and longer dendrite and neurites overall. So I think this is something to do with the disruption of this pathway. There is also another pathway that is related to increased neurite length growth, which is ERK pathway. And in the 16p deletion neurons, also ERK pathway is dysfunctional. So this one might be one explanation for this clear deficits between these neurons, between the control cells.
So does it mean that the 16p deletion cell type would be more functional, like better functionally compared to the control line, or not necessarily?
Not necessarily. Like, I think it's a disease phenotype because these patients have developmental disorders and increased brain growth as well. So macrocephalic brain growth and increased cortical tissue overall. So I think it's something to do with the proper differentiation. So maybe the cells and axons just keep growing uncontrolled manner because of the copy number variation that they are carrying.
Thank you so much. Yeah, this is good learning because I just think about it. The longer the axons, the better the neurons are. But in this case, not necessarily because it's a specific phenotype of disease. Yeah, thank you. I mean, just a common sense thing, but it's not necessarily the case. There are phenotypes, and this is cool because it's not always just smaller axons means disease. It can also be longer axons. That's super nice. Any questions from our audience or from online? Okay, if that's not the case, then I guess thank you so much, Maria. Please stay because we're going for the roundtable next. Thank you. Okay, so I have already introduced Danny, but I can reintroduce him again. So Danny McSweeney is from the PAC lab at the University of Massachusetts Amherst in the US. You have seen him a while ago, and he's also he's using the MaxOne system for characterizing the iPSC lines that they have in the lab. So Danny, are you now ready? Hello.
Hello?
Perfect. Okay, so
Okay.
I think your presentation is now up. Once you see it, you can start. Thank you.
Thank you. Hopefully the sound will work. First off, thank you so much for that introduction, Marie, and apologies for all these technical challenges. If there is anything wrong with my sound, please just let me know. And I want to say thank you to MaxWell for organizing and hosting this great event. I really appreciate this opportunity to talk. So my name is Danny, and I'm a graduate student in Dr. Chung-Hee Pak's lab at the University of Massachusetts Amherst in the US. And today I'll be talking about our recent efforts to investigate the neural connectivity and function of disease-associated loss of function mutations for one of our favorite proteins and genes called CASC using human-induced neurons. So in the POC lab, we study synaptic cell adhesion and neuronal signaling to better understand how these specialized structures guide neural connectivity in the developing brain. And so we're currently focusing on two distinct developmental time windows early in human development, including neuron formation or neurogenesis, which you can see at the bottom left of the figure on the left, and synapse formation, which is about halfway up that timeline. So neuron formation occurs very early in brain development, and it's an activity-independent process in which cells are differentiating and beginning to form their axonal and dendritic projections, which is summarized in the schematic on the right side. And then later, on the bottom half of that schematic, you see synapse formation taking place, which continues throughout life. This is an activity-dependent process involving a cycle of synapse initiation and specification followed by elimination, which then repeats. So these two processes are driven by proteins in the neuron and developing synapses, one of which is this protein called Casc. So Casc, or calcium calmodulin dependent serine protein kinase, is a membrane-associated guanylate kinase, or MAGUC, multi-domain scaffolding protein. It's implicated, it is, there we go. It is implicated in neurodevelopmental disorders like autism spectrum disorders and X-linked intellectual disabilities. Casc is expressed in both vertebrates and invertebrates, and it's especially highly expressed throughout the mammalian brain. The Casc gene is well conserved across mammals. On the right figure, you can see the rat Casc homolog and then the human Casc protein structure, and they carry about 98% homology between the two. So there are 6 distinct protein domains, each of which has sites for various protein-protein interactions. And examples of these interactions are listed along the top of that, or along the bottom of that rat homolog, although these are the same binding sites for human Casc as well. Depending on the developmental time window, Casc is found in several subcellular compartments where it plays distinct roles. And using the synapse cartoon on the left side, I'll walk you through some of these. In the presynaptic side, Casc binds to adapter proteins Mint1 and Veli to aid in assembly of vesicle release machinery, and it also interacts with N-type calcium channels and Neurexin-1, which is a cell adhesion protein that's required for efficient neurotransmission across that synaptic cleft, and is also implicated in neurodevelopmental disorders like autism and also schizophrenia. In the postsynaptic side, you can see Casc interacting with syndecan-2, which is another cell adhesion molecule, to regulate the stability and maintenance of dendritic spines. And Casc also forms connections with actin molecules, you can see kind of on the right side, and helps promote local microtubule assembly. Casc is also known to bind to the transcription factor TBR1 and translocate to the nucleus, where they regulate genes important for early cortical development and synapse formation and function, genes like relin, which is important for neuronal migration, which comes after neurogenesis, and the NMDA receptor subunit gene, NR2B. So Casc, to me, is a pretty remarkable protein in that it's found in multiple subcellular compartments and functions pretty differently depending on that localization, depending on its protein binding partners, and depending on the developmental time window during which it is expressed. There have been many Casc loss of function mutations identified throughout the protein sequence, some of which you can see running along the bottom of the human Casc homolog on the right. The clinical phenotypes that we see can range from mild to severe. This example here on the left is of a point mutation in the CAMK-like domain of Casc, and this pair of MRI scans highlights the structural malformations of a young boy's developing brain on the right with a Casc deficiency, paired with an age-matched control on the left. And in this scan, you can see pretty clearly severe microcephaly, or underdevelopment of the forebrain along that top, hypoplasia, or a lack of cells in the brainstem and cerebellum right in the middle where those white arrows are pointing. So the severity of Casc loss of function mutations can be quite harmful, and Casc mutations often also lead to cognitive differences, as I said, X-linked intellectual disabilities, autism spectrum disorders, and seizures as well. So given what we know about Casc's expression, subcellular localization, binding partners, potential role in both developing neurons and functioning synapses, we aim to answer sort of broad Casc questions, how it regulates early neuronal morphological development, and understand Casc function at the human synapse. So we take a multidisciplinary approach to answer these questions, including using these well-established transcription factor-mediated differentiation methods for producing human neural cell types from pluripotent stem cells, either from patients or engineered lines. And we're currently focusing on the human cerebral cortex, so we're utilizing these methods, which are listed on the right, for producing homogeneous populations of induced neural cells, both excitatory and inhibitory neurons, as well as human astrocytes. And for the purposes of my talk today, I'll be focusing on the blue cell, which is the cortical excitatory neuron along the top, which are driven by expression of the transcription factor neurogenin-2, or NGN2. So I'll refer to them as NGN2 neurons. We also utilize CRISPR-Cas9 editing to produce Cas knockout isogenic lines from a human male H1 wild-type embryonic stem cell background by first CRISPR editing, followed by selection, picking colonies, and we ultimately established 2 independent constitutive Cas knockout human embryonic stem cell lines called knockout 1 and knockout 2. You can see on the bottom left, there are these frame shift deletions in the beginning of exon 1, one line carrying a 14 base pair deletion and the other a 10 base pair deletion, both of which remove some or all of the start codon, which is in that red right there. So a complete loss of Cas protein was confirmed with Western blot, which is shown on the right there. And we're now able to use these differentiation methods on our wild type and Cas knockout ESC lines, introduce NGN2, and then produce homogeneous populations of human-induced neurons with the goal of analyzing our mature day 7 neuronal developmental time point, as well as our synaptic development time point, which is a mature day 28 time point. So these are just some representative images I wanted to show following the differentiation of wild-type and Cas knockout cortical excitatory neurons. The left panel showing images of our immature wild-type in the top row, followed by our knockout lines below. The markers listed on the top are tau and MAP2 to visualize the neurite projections, and then a merged image on the right with a nuclear stain in that right column. In the right panel, the rows are the same genotype, so wild type followed by the 2 knockouts, but this is our mature time point, our day 28 time point. And along the top, the first column is a synaptic marker, synaptophysin, followed by MAP2, showing the bodies of the cells, and finally a merged image. So we can successfully culture our neurons, have them reach both maturity developmental time points, and then this is what they look like. So to begin investigating how Cas may be contributing to early neuronal morphology and development, we first performed bulk RNA sequencing to quantify changes in gene expression for immature wild-type versus Cas knockout neurons. And about 900 differentially expressed genes were called from the bulk RNA-seq, with about even numbers of up and down regulated genes being represented. When we ran these DEGs through a gene set enrichment analysis, or GSEA, we found that there was very little enrichment in the downregulated genes, but a significant enrichment in the upregulated DEGs, which you can see here on the left. So this is just upregulated genes. And this is visualized using a cluster network software called Cytoscape, with each white hexagon representing a gene attached via a string to an enriched biological pathway or cellular compartment. So the more strings associated between genes and pathways or compartments, the more enriched that pathway is, for example. So there are 3 distinct biological pathways that were enriched in this upregulated gene set, which are represented in the varying blues. And we also found that several upregulated genes were identified as synaptic, which is in yellow. The 3 pathways being cell projection, morphogenesis, neuronal development, and cell adhesion. And then, so then you can see that there are 3 sort of main clusters of genes in the middle that are showing synaptic genes that are enriched in these multiple biological pathways. So then we performed a QRT PCR validation to verify what we were seeing here in the RNA-seq, and almost all of the probes selected produced significant readouts for differentially expressed genes, and with several of these genes identified as synaptic using the SynGO online database. So we feel that this is a pretty good first pass test to indicate that Cas in some way influences the regulation of gene expression at this immature time point. To follow up that study, we sought to quantify differences in neurite outgrowth morphology using immunohistochemistry and fluorescence confocal microscopy. So on the left, these are representative images of our wild-type neurons in the first column, followed by our 2 knockouts on the right. And below those raw images are binary mask overlays that we generated using Imaris imaging analysis software to quantify the neurite outgrowth. And we did this by measuring soma size, total dendritic length, number of branch points, and number of primary processes. And we found a significant overgrowth phenotype with the biggest difference in number of branch points and total dendritic length, with some upward trends in soma size and the number of primary processes. So this suggests that in day 7 immature neurons, Cas does regulate neurite outgrowth and gene programs important for this process. So moving on to day 28, our mature time point, we use the same immunohistochemistry and confocal microscopy methods and same parameters to characterize these cells. And we actually found that dendritic arborization was unaffected at this mature time point. We also saw no significant change in synaptic density, which was we measured along a fixed given length, nor did we see differences in synapse volume. And so despite synapse formation remaining unchanged at this mature time point, we still wondered whether Cas could be contributing to function. So we sought to measure these changes using both the MaxOne system that we are all very fond of, as well as whole cell patch clamp electrophysiology. And so we first started with the MaxOne MEA chips, and this is a schematic of our general, very straightforward workflow. I as well am not an expert in MEA. We got a lot of help to figure this out. And it was really a really fun experience. We started by plating a 1-to-1 ratio of excitatory neurons to mouse glia, which the neurons need to reach synaptic maturity, and then used, it was a PEI pre-coding followed by Matrigel for the final plating. Then we performed a full scan configuration using the activity assay to gauge a successful plating and identify those active electrodes. Then we performed our network analysis and measured changes in spike activity and burst patterns across that network. So these are some representative active area heat maps recorded for 2 of our time points that we were investigating. Day 21 is synapse formation has already started, and you can see activity. And then our more robust mature time point is the day 28, which is considered our mature time point. And these active areas are around 80%, so we're getting really good plating. And as a cutoff, we only included chips that had at least 50% active area. And so once electrodes were chosen from the full scan, we moved on to the network assay. And so here's a representative video. Let's see, I'm not sure how to play it. Is someone able to do that? There we go. Okay, so this is sort of a representative video of that random spike activity that we see, just random spontaneous firing across the array. And about 10 seconds in, all of a sudden, the entire array lights up with a burst, there we go, of synchronous firing. So this is indicative of simultaneous network signaling across the culture. And then after the burst, the culture quiets down and returns to that random spike activity. And then eventually, there we go, we see more of those bursts occur. So we used all the built-in Max Live software analysis metrics to characterize these changes in the makeup of the bursts, as well as the burst patterns across our 3 genotypes. These are 3 representative raster plots of our day 28 mature cultures, with wild type being along that top there and the knockouts below. We first quantified spontaneous activity across the array and saw a significant decrease in the mean firing rate and spike amplitude with an increase in the interspike interval. Then we measure the network, and these are just 3 examples of the differences that we saw, although we measured a bunch. We saw a significant decrease in burst frequency and percent of spikes within the bursts, as well as an increase in the interburst intervals. So we found quite a stark defect in neuronal signaling at the network level, and we hypothesized that this deficit may be due to defects in the coupling neurotransmitter release machinery and calcium channels at the active zone, which has been previously reported for our Cas protein. So to dive a little further into this question, we performed whole cell patch clamp electrophysiology. We measured a variety of things. One of the things that we did measure was differences in spontaneous postsynaptic currents in our wild type and Cas knockout neurons to gauge the validity of this hypothesis. And what we found was a significant decrease in the frequency of the spontaneous iPSCs. It was a decrease by about half, with no change in the amplitude. So this seems to suggest a defect in the presynaptic release probability due to this Cas knockout. So given these electrophysiological phenotypes, we then investigated the transcriptomic changes associated at this mature time point using bulk RNA-seq. The workflow is similar to what we did for day 7. About 1,700 differentially expressed genes were called, with about even numbers of up and down regulated genes, which you can see represented in that volcano plot on the left, with the down regulated genes being in blue and the up regulated DEGs being in the red. And so quite a number of these DEGs that were called had already been mapped in the SynGO database as synaptic genes, and many of which were associated to pre and postsynaptic function, either pre or postsynaptic function, like the 2 groupings shown there in the long rectangles. The graphs along the bottom are representative fold change values for several DEGs that we found interesting. These are for our 3 genotypes across 4 technical replicates that we submitted for RNA sequencing. And there are several significant DEGs for genes that regulate synaptic vesicle release. There are genes that are postsynaptic cell adhesion molecules, some of which are listed here, including GRIA3, CBLN2, SLIT-RK3 is another one. There were DEGs for genes that were neurotransmitter receptors, signaling molecules, just to name a few. So similar to our immature time point, we then used GSEA, the gene set enrichment analysis, and cluster network software to visualize the enrichment of biological pathways and cellular compartments in our mature data set. And we did this for all DEGs, so this cluster network includes both up and down regulated genes to get a general view of dysregulation in gene expression programs. Again, all the hexagons, both the black and the white, are representative DEGs, and the rectangles being pathways or compartments. There were 7 major biological pathways in varying colors of blue. All the blues are pathways, and then 6 distinct cellular compartments enriched for this data set, which are in varying colors of red. And the colors do not denote any sort of intensity in dysregulation, just clumping pathways with cell compartments. And then, so some of the major pathways affected are synaptic signaling along the top, cell projection morphogenesis, cell junction adhesion, and so on, as well as enrichment in both the pre and postsynaptic compartments. And then the black labeled cluster of DEGs in the middle, we found were enriched in all or almost all of the pathways and cellular compartments, and many of these genes in the middle mentioned on the previous slide are found in this central cluster. So this provides a really interesting and broader snapshot of how loss of Cas as a scaffolding hub in synapses may be sort of globally influencing a neuron's signaling programs. So we concluded based on these findings that in immature neurons, CASC regulates neurite outgrowth and gene expression programs that are important for neuron development. And in our mature neurons, CASC mediates synaptic function and neurotransmitter release probability. Some future directions, although we have many future directions we would like to do, are we'd like to look at excitatory inhibitory balance and whether CASC is somehow contributing and using a mixed neuronal culture, having both excitatory and inhibitory neurons in a single dish. We'd also like to investigate human binding partners of CASC across developmental time windows and then measure activity-dependent changes to that interactome. Lastly, I wanted to note we've submitted this a manuscript with these findings for review, and you can also find a copy on bioRxiv. The link is in the bottom left. I believe there may be a link being shared in a chat as well. And if you'd like to reach me, please feel free to reach out. Email me anytime. My email is right there, dmcsweeney@umass.edu. So this project wouldn't exist, nor would my job without my wonderful PI, Chang-Hee, who has mentored me for 3 years, 2 of which are in a pandemic, and has answered every single possible question I could think of with such patience and enthusiasm. Thanks to my entire lab for such amazing support, and our collaborators at Rutgers, Jiping Pang and his grad student Le for mentorship on electrophysiology, Bruce Aronow and Kong out of Cincinnati for RNA-seq analysis, and then Jim Chambers at our microscopy core for a ton of helpful training. I just want to say a huge thank you to Marie, the marketing team here, the technical support team during this whole process at MaxWell has been amazing. Thank you for hosting such a great event and providing a ton of help over the last few years with our MaxOne system. I do want to give a special shout out to Giulio Sorsi for being a fantastic person to work with. He's provided unending support both remotely and on site. Thank you all for attending, and I'm love to answer some questions.
Thank you so much, Danny. Yeah, congratulations for getting all of these results during pandemic period. This is really amazing. Okay, so do we have any questions for Danny from the audience? Yes, we do.
So again, it's me, but I have really I'm very curious about one thing. So you combine basically measurement of the MEA, basically network activity, and then you also have this wholesome patch clamp results. And if I remember correctly, you mentioned from the patch clamp result that you infer that there is maybe a reduced probability of synaptic release. And I was wondering maybe also your thoughts about trying to combine these findings. So from the network assay, you can basically infer the overall activity of the, of the entire network. And maybe if you combine that with the single cell patch clamp, you can even, get one step beyond. So just would like to have your thoughts on that.
Yeah, definitely. I mean, I think that's a great approach for sure. I mean, I feel like some of our data with the reduced firing rate from the MEA, it does agree with our synaptic release machinery hypothesis being a defect. And yeah, I think I've really enjoyed the two. So those two techniques are very new to me. We started patching as of last summer. And so I'm definitely still learning how to integrate all of these technologies that we're using, but I think using them synergistically like that is a great idea, and I would love to dive further into the MEA data. We use the MaxLive software to do sort of a first pass analysis, and we worked a little bit with Elena at MaxWell to actually use the MATLAB code to dive in further. So my hope is to sort of tease that apart more using both MEA and patch.
Let's just continue with this discussion, of course, after this event, because I think this would be very interesting, not just to, combine these different thoughts. Thanks, Danny.
Yeah.
Thank you, David. Now we have some questions from our online audience. This is from Mark Hester from Nationwide Children's Hospital. Great talk, thank you, according to Mark. So he was curious if you had tried differentiating your cask knockout iPSCs without transcription factor-based induction methods, for example, using small molecule-based differentiation methods.
We haven't. We haven't gone down that road. I've mostly used the NGN2 protocol. I've started using the other two as well, and I believe some of my colleagues are trying out some different differentiation methods. We did try a I think it was a dual SMAD inhibition protocol in tandem with our differentiation methods to sort of help drive the neuronal fate. But we haven't exclusively gone down the road of small molecules. Yeah.
All right, thank you. And I think you may have seen some talks earlier and also yesterday. Are you also interested in using the axon tracking assay to characterize the differences between the wild type and the knockouts?
Yes, I'm very excited to. That's basically at the top of my list. Julio and Martina from MaxWell visited maybe a month ago, and they showed the axon tracking. It was gorgeous. It was a really cool assay. So yeah, I am very excited to integrate it.
Okay, thank you so much. Yes, I think the rest of the questions you'll ask later at the round table. Okay, thank you.
Thank you.